activity in iPSC-derived excitatory neurons. So I'm going to orient us now on how our lab fits into CHAMP1 research as a whole. And we know that CHAMP1 is critical for mitosis and DNA repair mechanisms, as others have shown earlier today. But we know very little about CHAMP1's role in human neural lineage cells. And we know that this is critical work to be done in a human model to capture the human-specific mechanisms of mitosis and neuronal maturation. So I'm going to be looking at the question of how CHAMP1 modulates human neurodevelopment. To do this, we are using a model of human neurodevelopment using patient-derived iPSC lines. So this process begins here with the blood samples from patient donors, and these blood cells are reprogrammed back into this pluripotent state, so much like a blank slate of a cell. And our lab acquires the cells at this iPSC stage. These will eventually become differentiated into mature neurons. From there on, we can perform our experiments. And what we really want to capture in this study is a range of CHAMP1 properties, which is why we included one neurotypical donor line, which I'll refer to as control. We have two patient-derived iPSC lines, and I will be referring to them as CHAMP1A and CHAMP1B, just for sake of time. We also included an isogenic knockout, so this is a total knockout of CHAMP1 from a neurotypical background. So here I'm showing you the steps taken from iPSCs to become mature neurons. We use different chemical compounds that promote the change in cell identity from the stem cell phase into these neural progenitor cells. Our lab is interested in this dividing phase in the neural lineage, but today I'll really be focusing on the neural maturation phase, both at this earlier time point and then a little bit later in the process of neural maturation. But what's really interesting is that these iPSC-derived neurons in culture, they do develop similar structures and functions as neurons do in the developing brain. So like I said, most of the data I'll show you today is focused on these two time points during neural maturation of kind of early neurons versus a little bit later. So I just want to show you, what this actually looks like in the lab. So these are neurons at DIV 30, so day in vitro 30. And I want to draw your attention to these really beautiful green cells. So these are neurons in culture. And at about 1 month, so DIV 30, they start to develop these physical features that are very similar to what we see in living brain neurons. Specifically, they develop structures called dendrites and axons, and these are critical for receiving information from other neurons and sending out information. So we see that they do develop these physical structures similar to the living brain. They also, starting relatively early around DIV 30, they can start sending and receiving electrical signals. And these are also neurons. It might be a little harder to see than the slide before. It's a bit more zoomed out, so there's more cells in this picture. But what I wanted to highlight is really how complex this network is becoming. Even though these are all the same cell type, they're starting to develop a functional network within just a piece of glass growing in a dish. So they're really fascinating to work with. You have differences in the culture between some neurons that are more flat and kind of spread out in their processes, and then you have kind of little hubs of many, many neurons clustered together and talking to each other. So we're using this model to answer two questions, and the questions I'll be focusing on today is, do these mutations in CHAMP1, assuming it's a loss of function mutation, do they change the structure of the neurons? And to do that, I'll be using this process of immunohistochemistry. So all of these brightly colored pictures that you're seeing are samples of neurons using immunohistochemistry to label for certain markers. The second question is looking at, does CHAMP1 loss of function disrupt the neural activity, so the function of the cells? For that, I'll be using electrophysiology, which I'll get into a little bit later. So again, all of these experiments are focused on these two developmental time points because this is such a critical gene for neurodevelopment. We wanted to have at least 2 time points to really compare over time. And so I first looked at the structure using different markers of genes in these neurons, and I really wanted to capture how their structure was changing over time, as well as using a functional analysis using electrophysiology. So what do we expect to see in these cells in culture as they're growing? We know that neurons, over the process of maturation, they grow larger. Their cell bodies or their somas get bigger. They also get more complex. Their branching starts to extend. And they also start to gain these functional electrochemical properties as they mature. So these are all properties that we can screen for in this model. And we can compare that across lines and across time points. So I'm going to focus first on this question of the structure of neurons related to CHAMP1. And what I'm showing you here is individual neurons that I labeled with a marker that fills the cell body and all of the cell branches. So you can see from the 4 lines included here, these are all taken at DIV30, and you can really see some differences in the branching of the neurons. We also looked across at DIV60 as well to try to capture changes over time. But the main difference I wanted to point out is, especially in the CHAMP1 isogenic knockout, we see a reduction in the complexity of these branches. You can really tell here between these 2 examples, as well as compared to the control. So how do we quantify these things? We took these images and we just traced them, and we're able to reconstruct the branches and then quantify not only the length of these individual branches, but where they were in relation to the soma, because this can tell us more about the functional properties of the neurons. So I'll get into some of the highlights of that data now. So I first looked at DIV30 for just the total length of all of these neurites. And I did see some downward trends in the CHAMP1 mutant lines, but really the most striking difference was here at DIV60. And this is in the knockout compared to the control. So this change in the length of the neurites could represent some deficits in mechanisms that are important for signaling between neurons and potentially for the migration of these neurons during development. I also looked at where these little branch points or these intersections were in relation to the location of the soma. And what I wanted to really focus on is that there were some subtle differences at DIV30, but again, the most striking difference was in the location of these intersections in the CHAMP1 knockout. They were more clustered around the location of the soma compared to the control. And there was some changes too between the CHAMP1A group as well as the CHAMP1B, although it wasn't quite significant. But again, what this could represent is that there might be some deficits in the growth mechanisms or in some signaling mechanisms that are important for neuronal migration. Okay. So I've shown you so far that there are definitely seen in culture some physical growth changes as it relates to CHAMP1. And next I'm going to look at the activity and functions of these neurons in vitro. So during development, we know that neurons become increasingly more electrically charged, and they're able to fire action potentials. So you might have heard of action potential before. It's this classic shape of an increase in activity. It goes up and down, and what this represents is this very fast transient change in the voltage of the cell. It starts here at the cell body, and then that action potential travels along the axon all the way to the axon terminals. And when this action potential hits the end of the neuron, this is where the neurotransmitters are released. So this is a truly foundational aspect of how neurons communicate with each other. But I focused on properties of these action potentials, both their shape and their frequency, to really describe the functional output of these cells in culture. So how does one go about doing this? There's different ways that we can study this, but I focused on a method that is highly specific to looking at an individual cell at a time. This is called whole cell patch clamp electrophysiology. I'll call it EPHIS for short. But what I do is I take a very small glass pipette that is filled with a very salty solution. I'll talk about in a minute that neurons are very salty. But this glass pipette will physically adhere to the cell body of one cell at a time. So I'll apply some suction, and then I'll break into the cell membrane. And this allows me to connect the pipette with a whole computer setup. I can then manipulate properties of the cell and compare these functional properties across many cells at a cell-by-cell basis. So this is just a picture of the electrophysiology rig. You'll notice some potions on the side. But what I'm able to do is keep the cells alive on the microscope by incubating them in sort of a Gatorade-like solution. And this is a picture of an individual cell that I patched with a very tiny glass microelectrode, and it is adhered right on the cell body. So we'll first start by showing you data of individual action potentials and show you how this is changing with CHAMP1 mutations, especially in the shape of these waveforms. So I'll point your attention to this single action potential here in navy. This is the control. We then have the CHAMP1A in the light blue compared to the CHAMP1B in orange, and then this is the CHAMP1 knockout in pink. And if you compare these to the control, you can already see some shifts in the shape of these waveforms, mostly in where their peak amplitude rises to and in how they repolarize. So I'm going to zoom in on this a little bit to show you just that change in the shape. Here again, there's the control action potential, a CHAMP1A, a CHAMP1B, as well as the knockout. And so overall, I'm seeing these changes in the shape of the waveforms. So what this means is that when we see waveforms that look like this, it shows us that they're likely more of an immature cell. They haven't fully gained those properties that allow them to have strong action potentials. So that is definitely what I found at DIV 30 through 40. I then looked again at DIV 60 through 70, and I saw that a lot of these properties did improve over time. So these waveforms are starting to look more like the control. But I still noticed that there was sort of a shift or a separation between these two groups, the CHAMP1A and then the knockout, were still more different than the control than the CHAMP1B, whereas the CHAMP1B looks much more similar to the control at this time point. So what all of this means is that this could reflect that there is a delayed maturation timeline. It can mean that there is a physical deficit somewhere within the neuron and the mechanisms required for action potential firing, or it could be an interesting combination of both of these things happening at the same time. So to look further into this, we know that in the living human brain that mature excitatory neurons have to fire trains of multiple action potentials in order to encode information. This is another one of the foundational properties of how neurons work in the brain. If you think of it like a computer that's encoding with zeros and ones, the ones are the action potentials and they need to go really fast. So these neurons have to acquire this property for highly repetitive action potential trains, and we know that in culture that these neurons are able to do this, so we can use this as a model and compare across lines. But what you can already notice is along with some of these changes in the shapes of these waveforms, we are starting to see that there's differences in how frequent these events are. So I quantified this here in an experiment where I incrementally increased the amplitude or the input into the cell. So I'm giving the cell a stimulus from 0 to 100, and I'm asking how many action potentials is going to come out of the cell. We also call this an input-output curve. But if you look at the control line up here in navy, you get increased action potentials up to around 8 hertz, and then they do start to kind of trail off here. But most importantly, we see that there is a reduction in the overall frequency across amplitude steps in all of the CHAMP1 related lines. I looked again at DIV 60 through 70, and all of the data is kind of merged on top of each other, so it's a bit hard to see. But what I saw is that the CHAMP1B in orange is looking much more similar to the dynamics of the control in navy, whereas there's still a reduction in the overall frequency in the knockout here in pink, and then the control A line. So what this tells us is that at this very early developmental time point, that there's a change in how these neurons are responding to stimulus. So I wanted to step back and say, okay, how does repetitive action potential firing, what does this mean on a population level? So I looked at all of the neurons that I recorded from at DIV 40 and DIV 60 in all 4 of the groups, and I asked the question, is this neuron firing multiple action potentials, or is it most mature, and then is it only firing a single one, a failure, so there's a little bit of a peak but not really classified as a real action potential, or is it not firing at all? And so I used the scale to classify these neurons individually. And what you'll see here is that in the control, most of the population are these very mature looking repetitive action potential firing neurons. In the 2 CHAMP1 patient-derived lines, we do see that shift in the population. So this darker color here is representing those most mature cells, but there are some that are only firing single action potentials. When you look within group, so between this row and this row, what you're seeing is a change in the population over time. So more cells are acquiring that property of firing many more action potentials. The same for CHAMP1B, and there is just some subtle differences here between groups. But what was most strikingly different was in the knockout. At DIV 30 through 40, most of these cells are only firing single action potentials. So this is a very clear indication that there is likely some delay at the population level, but even in the knockout, there was a shift. There was growth happening over time. Okay, so in order for all of these properties to happen, neurons have to have functional ion channels, and as neurons develop over time, they grow both in their size and in the density of ion channels that are docked at the membrane. And what ion channels are is basically little doors that will open and close to certain ions based on the voltage that will unlock or close the door. And so action potential firing is highly dependent on both sodium and potassium. So we're going to be looking at these 2 ion channels. And I find it helpful and also a little funny to consider neurons like a salty banana. So if you want to remember the direction of these ion flows, the neurons have lots of potassium on the inside of the neuron and lots of salt on the outside. And these ions want to move against that concentration gradient. So the potassium wants to leave the banana, the salt wants to come in. Okay. But all of this is relevant to us because we know that deficits in ion channels on the neuron have been associated with other autism spectrum disorders. So this is a critical piece of the puzzle to look at in a potential therapeutic target. So what I did is I looked at how the ions moved in and out of the neuron, And I'm just showing you a raw trace of that electrical information. And what I'm seeing is that sodium will always go into the neuron very fast in this negative direction, and potassium will always go outward on a slower phase. And with this information, I can record from neurons and plot how dense the ion channels are on the membranes with increasing steps. So what you're looking at here is a plot of sodium coming into the neuron and then potassium going out of the neuron. And this is just at increasing voltage, so I can look at the dynamics of the cell. And what I'll draw your attention to is that the control in navy is much more negative on the scale of inward sodium currents. So there's a lot more sodium ion concentration in the control compared to all three groups. And that's also true in the potassium ions, but it's a little bit more close together. I looked again at DIV 60 through 70, and what we found was pretty striking, whereas the control cells in the CHAMP1B clustered much further away than the CHAMP1A and the knockout, both in the sodium channel area and also in the potassium channels. When I compared, I've just shown you these two data sets, and when I looked at them across time, you'll see that the control in navy and the CHAMP1B in orange, they get more dense over time, so more negative in this direction. So that means that they are increasing in their ion channel density over time. However, the CHAMP1A and the knockout did not seem to really change much between these two time points. So what this is pointing to is that there might be some deficit in, there might be a delay in the process of how the ion channels are accumulating at the cell, or this process might be stalled at a certain critical time window. I next was just curious about, is this related to just how big are the cells? So typically neurons are going to grow both in ion channel density and in size at the same time. So if you have a bigger cell, you're going to have more ion channels. So I looked at a measurement of cell size, which is membrane capacitance. So larger numbers mean bigger cells, and more negative numbers here on inward sodium mean more ion channels. So I'm expecting to see a correlation in this direction. And I do see that in control. So bigger control cells do have more sodium channels. I only saw this correlation at DIV 30 and 40 and DIV 60 and 70 in the control group, but the CHAMP1B was trending towards that significance, but this was not significant in either of those groups. So what does this mean? We think it could mean that there is a disruption in the accumulation of ion channels regardless of how the cell body is growing in size. Okay, so we've looked at kind of activity properties of individual neurons down to their ion channels. The next question is, how are these neurons talking to each other? And so the communication between neighboring neurons happens at the synapse. This is a really small space in between neurons that allows for signals to pass between them. These electrical currents are very small, and what they represent are these tiny little pockets of neurotransmitters, and they're released into this synapse space, and they bind to the receptors. So like in the human brain, we can model this in culture. Neurons in culture do produce these spontaneous synaptic events. And important to us, changes at the synaptic level have been observed in other autism spectrum disorders, in other neurodevelopmental disorders, and it can inform us about how the network of cells is communicating. So my first question was at the population level. I just asked the question, is a neuron receiving synapses from other neurons or not? The darker colors in these pie charts represent the percent of the population that is receiving synapses, and then the lighter color is not. So in the control, you can see a large amount of the population is receiving synapses at both time points. The CHAMP1A and B have differences between each other, but they are increasing over time. So CHAMP1A going from 25% up to close to 50%, whereas CHAMP1B starting almost at 50% going up to 77%. But interestingly, in the knockout, the percentage of cells receiving synapses didn't change. It actually slightly went down. So this suggests that there could be a lack of connectivity within the population of cells, and something that we definitely want to explore further with other measurements of network activity. But I next looked at the synaptic events themselves, and I can measure, number one, how frequent are these events? So you're looking at, this is sort of just baseline noise activity of the cell, but all of these negative deflections are individual synapses, synaptic currents happening. So I'm counting how many synaptic events happened, and then the amplitude, how large are these events, or how small are they? So this is the synaptic current data across the 4 lines. This is just a snapshot at the first time point, but what you can probably already tell is that there is a reduction in the frequency of these events, specifically at CHAMP1A and in the knockout, as well as in the CHAMP1B. So overall, there does seem to be a reduction in frequency. However, when we looked at the amplitude of events, I think this was only significantly different in CHAMP1A and knockout compared to control, whereas CHAMP1B was fairly similar. What was interesting about this is it seemed to get better over time. So the amplitude of the synaptic currents at the second time point was more similar across groups, and the only change in frequency was in the CHAMP1 knockout. Let's see. So taken together, there might be some role for CHAMP1 in regulating synaptic maturation, both at the individual neuron level and at the population level. Okay, so just to highlight some of the results I've shown you, with CHAMP1 mutations in neurons in culture, we've seen changes to physical properties, how the neuron responds to stimulus, and also synaptic connections between neurons. What does this all mean, especially for all of us in CHAMP1 research together? What it means is that we can screen many different iPSC-derived neuron lines for both changes that are occurring because of CHAMP1 mutations, and we can also use this model to look at functional responses to treatment in vivo, or in vitro. So overall, some of the conclusions is that we did find mutation-specific delayed neuronal maturation. We saw differences between 2 of the patient-derived lines as well as the knockout. We saw deficits that were observed both at the single neuron level and at the population level. And this might point to a potential function for CHAMP1 in these post-mitotic neurons. So future directions for me in particular in this project, I'm really interested in looking at functional assays at the population level using a different technique called a multi-electrode array. This will allow me to record activity from many, many neurons at the same time, and I can look at, are they firing synchronously? Are there, any changes at that level? As well as I want to look at the morphology of dendritic spines. These are really small structures, but this has also been associated with some other disorders. The lab as a whole is very interested in single-cell genomics to look at transcriptional changes with CHAMP1 as well as translational approaches. So with that, I'd like to thank everyone in the lab. Most of our lab is here, this amazing team. So thank you to them, and thank you for the foundation for letting us be here today. So I'll take questions now.
Thank you very much, Dailey. Questions?
this is really interesting. Did you try to increase sodium conductance, membrane conductance, or decrease potassium conductance and see if this could increase the expression of voltage-gated channels?
No, we haven't tried that. We really have just started with doing sort of a panel of functional assays, but I'm very interested in looking more at the sodium channel level, especially looking at different subtypes of sodium channels. But yeah, that's a good question.
Did you study, do you have any single channel recording?
No, I don't, but I would love to do that. Yeah, that would be really exciting. Yeah, we have the technology to do that.
Hi, I'm just a parent, so if this question is too far from what was explained, let me know. So thinking about all of our kids who are in all their therapies and what you shared and neuroplasticity, like, is some of this correlated to the dosing of therapies? Is there hope for is it better to do higher frequency, shorter time, or more time? Or is what you studied connected to the neuroplasticity we might hope for in therapy?
That's a really good question. I mean, I think it's all very connected. I will say with this model, we are looking at a very specific developmental window in one type of cell. And so looking at the whole brain, there's different types of cells that work together as a circuit. I think with looking more at the synaptic level, we know a lot of plasticity happens at the synapse level, and so I'm really interested in looking more into that to see how these cells respond maybe to stimulus or other ways that we can model learning, or at least changes at that level. So yeah, I would like to look more into that.
So here's a question from one of the doctors watching virtually, and I've actually heard this from various diseases, but any of the parents in the audience, just show of hands, crave salt? Would any parents see their kids crave salt or crave salt? One hand? My son liked salt on everything, liked olive oil and salt on everything. I don't know if that's a taste preference or it's related.
That's really interesting. I think that might be more of a question for sort of the gut-brain axis, which is a really interesting area, but I don't think it is a direct correlation to sort of ions at the neuron level, but definitely getting appropriate amounts of dietary supplements with different ions is, I think, important just for general brain health. But yeah, that is interesting.
So I've got a question. So a lot of our kids with CHAMP have really complex medical symptoms. So like my son's been on, has reflux really bad since he was born, and they put him on Prevacid, long term, and I've been researching the impact of that on absorption of potassium or different things. He also has a sensitivity to bananas. It actually triggers a histamine response, and then he has a cyclical vomiting. So how do these complex medical symptoms impact this neurobiology? He's probably not getting enough potassium because he has these different things. Have you guys done any research into that on the effects of these other medications or foods that go with our complex kids?
That is so fascinating. No, we haven't, but we do. I think one of our hopes is to, with a more higher throughput screening process, to look at least at drug treatments for individual cell lines and see any responses, especially if it's more specific to different ions. I think that would be really interesting to do. I have no idea about food and dietary changes, but I think that it's also fascinating. Yeah.
Any other question? Cool. So thank you very much once again.
Thank you, everyone.